class, and welcome to the last module in this course. Today we'll be talking about temperature stress combined with drought or salt stress. In this lecture we will go over a quick introduction, after which we'll explore drought and salt stress individually. Then we'll look at drought and temperature stress, and then a combination of salt and temperature stress. At the end we'll go over the summary as usual. Among the great number of individual abiotic stresses affecting plants, drought and salinity are the most severe. They limit plants' growth and crop productivity in agriculture worldwide. These two stresses could have damages exceeding the sum of that attributed to all other natural disasters, and when they happen together, they promote devastating changes in plants subjected to them. Plant responses to drought and salt stresses have much in common. The water limitation and water stress that accompany salt stress and drought is very difficult for plants to tolerate and maintain its normal growth and development. Recent studies have revealed that response of plants to combinations of two or more stress conditions is unique and cannot be directly extrapolated from the response of plants to each of the different stresses applied individually. Additionally, the simultaneous occurrence of different stresses results in a high degree of complexity in plant responses, as the responses to the combined stresses are largely controlled by different and sometimes opposing signaling pathways that interact and inhibit each other. So now let's have a look at drought. What is drought stress? Meteorologically, drought is a period without significant rainfall or a deficiency in water supply. In terms of plant physiology, drought is defined as insufficient water to meet basic needs when water in the soil is reduced and atmospheric conditions cause continuous loss of water by transpiration or evaporation. Water stress is considered to be a moderate loss of water, which leads to stomatal closure and limitation of gas exchange. Desiccation is a much more extensive loss of water that can potentially lead to gross disruption of metabolism and cell structure and eventually to the cessation of enzyme catalyzing reactions. When we talk about water stress in general, we think of reduction of water content and reduction of total water potential, which can lead to wilting and closure of stomata. But when we're talking about severe water stress, this may result in arrest of photosynthesis, disturbance of metabolism, and finally death. In this figure, we can see some classic signs of severe drought stress: footprinting, wilting turf turning a darker color, and severely stressed straw-colored turf. The thatch layer and underlying root zone will probably show signs of hydrophobicity. When faced with drought stress, plants respond with some physiological responses, which include osmotic adjustment, stomatal closure, leaf abscission and reduced leaf growth. This can help by reducing the surface area for water loss, increased root growth, which helps with looking for deeper water and other water supplies, increased wax decomposition on leaf surface, which reduces the cuticular transpiration and increases reflection, and finally reduction of photosynthesis. These responses improve the water use efficiency of the plant on the short term. Drought tolerance can be defined as the ability of the plant to cope with reduced water potential of the plant tissue. In order for plants to tolerate drought, plants evolved mechanisms of tolerance. Those are drought avoidance. Those mechanisms aim to limit transpiration or promote water uptake such that the plant tissue does not experience reduced water potential or dehydration. Examples on this are stomatal closure and the increased root to shoot ratio. The second mechanisms are drought escape. Those are mechanisms that allow the plant life cycle to be completed before drought reaches a critical level. Examples on this would be the acceleration of flowering and seed set. This is a basic figure that shows the initial reaction or the short-term adaptations of plants to water stress. Stomatal response, ROS scavenging, metabolic changes, and photosynthesis are all affected when plants are subjected to water stress. These collective responses lead to an adjustment in the growth rate of plant as an adaptive response for survival. In this figure shown in front of you, you can see the long-term or acclimation responses to drought stress and the short-term or the adaptation responses to drought stress. The majority of traits related to plant drought adaptations, as example, root size and depth, hydraulic conductivity, and seed storage reserves, are associated to plant development and structure and are constitutive rather than stress-induced. In addition, a large part of drought plants' resistance to drought is due to their ability to get rid of excess radiation. Plants can also endure water drought conditions by avoiding tissue dehydration while maintaining tissue water potential as high as possible or tolerating lower water potential. Dehydration avoidance is usually observed in annual and perennial plants, which has been associated to proper characteristics, as example, capacity to minimize water loss and maximize water uptake. The water loss could be done by stomata closure, reducing light absorbance by leaf, or through reflectance increasing due to the development of trichome layer. Or, on the other hand, the maximizing water could be reached by increasing in root systems, which is a result of alterations in allocations of plant resources. And now let's have a look at salinity stress. Salt stress is defined as a condition where excessive salts in soil solution cause inhibition of plant growth or plant death. Saline soils significantly reduce plant growth and crop yields. It is estimated that 6 to 10% of total land surface of the world is affected by salt. On a world scale, no toxic substance restricts plant growth more than salt does. Among the various sources of soil salinity, irrigation combined with poor drainage is the most serious, because it represents losses of once productive agricultural lands. The reason for this so-called secondary salinization is simple. Water will evaporate, but salts remain and accumulate in the soil. The stresses created by a high salt concentration in the soil solution are twofold. Many of the salt ions are toxic to plant cells when present at high concentration externally or internally, which causes the primary stress. Typically, sodium chloride constitutes the majority of the salts. Sodium ions are toxic to most plants, and some plants are also inhibited by high concentrations of chloride ions. Second, high salts represents water deficit or osmotic stress. Growth is immediately reduced when osmotic stress is present. General symptoms of damage by salt stress are growth inhibition, accelerated development and senescence, and death during prolonged exposure. Growth inhibition is the primary injury that leads to other symptoms, although programmed cell death may also occur under severe salinity shock. Salt stress induces the synthesis of abscisic acid, which closes stomata when transported to guard cells. As a result of stomatal closure, photosynthesis declines and photoinhibition and oxidative stress occur. An immediate effect of osmotic stress on plant growth is inhibition of cell expansion, either directly or indirectly through abscisic acid. Ionic stress occurs when sodium and chloride accumulate to toxic levels in the tissue. This process takes time to occur. The degree of damage from ions depends on the rate of accumulation and how effective ion compartmentation is within the cell. Here's a schematic view on salinity effects on plants. A general plant response to salinity effects leads to signaling, changing in gene expression, changes in protein expression, changes in plant metabolism, and affected responses leading to elevation of adverse effects of the stress. In order to respond to these stresses and survive, Plants undergo different responses based on the osmotic effect or ionic effect. Osmotic stress causes plants to respond by osmotic adjustment, which happens by accumulating ions, solutes, and organic compounds. Plant cells work best if they have the correct temperature, water levels, and ion concentration. Even though those environmental factors cannot be controlled by the plant, plants strive to adjust internally and create a state of homeostasis within the cells. Ionic stress causes plants to respond by creating ion homeostasis. Homeostasis can be defined as the tendency of a cell or an organism to maintain internal steady state. Here we can see some adaptive strategies for salt-tolerant plants that are trying to reach ion homeostasis. Ion homeostasis is mainly achieved through the regulation of membrane proteins involved in ionic transport. The main adaptive strategies of salt-tolerant glycophytic plants exposed to salinity are avoidance through ion exclusion, where plant roots must exclude most of the sodium and chloride dissolved in the soil solution and escape from gradual buildup of salt in the shoots to toxic levels, potentially through low membrane ion permeability. Second is tolerance, which is achieved through ion inclusion and possible compartmentalization into the vacuole. And third is osmotic stress tolerance, which enables the plant to remain functional despite internal ionic stress. This is primarily due to the accumulation of different types of organic particles such as soluble sugars, free amino acids, and free proline. Before we move to the effects of combined stresses together, let's go over a brief summary of the major physiological stresses when affecting the plant individually. As you remember, high temperature stress denatures proteins and causes lipid peroxidation. High light causes production of excess excitation energy in the photosynthetic reaction centers, resulting in direct accumulation of a variety of activated oxygen species. Water deficit or drought interferes with metabolism. ROS produced under drought conditions trigger signaling pathways that generate defense responses. And as you remember, cold stress interferes with metabolic processes, particularly enzymes activity, and alters membrane properties. Freezing or frosting can severely damage tissues when ice forms. Extracellular ice formation also causes intracellular water deficit. Soil salinity is usually caused by excess salts of chloride and sulfate. Salinity results in ion cytotoxicity and osmotic stress and decreased uptake of nutrients, resulting in metabolic imbalances leading to oxidative stress. Abiotic stress factors had been studied extensively. However, not as many studies had been conducted on combined stress effects on plants. In this stress matrix, we can see different combinations of potential environmental stresses that can affect crops in the field. The matrix is color-coded to indicate stress combinations that were studied with a range of crops and their overall effect on plant growth and yield. The red squares show potential negative interaction, the blue ones show potential positive interaction, the whites have unknown interaction, and the black have no interaction at all. If you look closely, you can see that drought has a potential negative interaction with salinity, heat, and chilling. From an ecological point of view, plants are usually not exposed to one stressor at a time, but rather multiple stressors. The resulting effect of multiple stresses can follow one of four patterns: additive, antagonistic, synergistic, and potentiation. The easiest way to explain these responses is by drawing an analogy to drug dose effects that is illustrated in the graph here. We could have two drugs, say A and B, and each one of them would have a certain effect at a certain dosage. If we would administrate half of the dose of the two simultaneously, we might get one of the following effects. Additive is when the two complement each other and the sum of their effect is the same as the effect of one whole dose. The additive effect usually indicates that the two pathways are independent and do not interfere with one another. Antagonistic effect happens when one drug somehow inhibits the other. In this case, the sum of the two effects is less than the whole dose effect of either one of the drugs. This clearly shows that there is an interaction or there is an interference in the two pathways. Synergistic is a case where the effect of combining the two drugs is amplified beyond the sum of the two. The last pattern is potentiation. This pattern is different from the others in the sense that one of the drugs will not trigger a response on its own, but in the presence of the other drug, it would produce an augmented response that will exceed a twofold magnitude of the other drug. The most common patterns found in stress effects are the additive and antagonistic. One of the reasons for small number of combined stress studies is the standardization challenges that should be applied to the experiments. The quantification and categorization of the outcome responses can be hard to evaluate in a field setting due to the uncontrolled interactions of other biotic and abiotic factors. Growth chambers and greenhouses provide control environments that can control the stresses applied to the plants, yet the results of these experiments may not be 100% representative to what happens in real life. Field experiments are the closest to what really happens in nature, and responses and results can be the most valuable, especially that fields can accommodate large numbers of plants, providing a significant assessment of phenotypes and stress effects in any crop. Salinity and drought stress show a high degree of similarity with respect to physiological, biochemical, molecular, and genetic effects. During drought, a finite amount of water can be obtained from the soil profile by the plant, causing ever-decreasing soil water potential. In most saline environments, a large amount of water is at a constant but under low water potential. Plants have a chance to adjust their osmotic potential, which prevent loss of turgor and generate a lower water potential that allows plant to access water in soil solution for growth. Physiological drought occurs when soluble salt levels in the soil solution are high enough to limit water uptake due to low water potential, thereby inducing drought stress. The major differences between the low water potential environments caused by salinity versus drought is the total amount of water available. Drought and/or salinization are manifested primarily as osmotic stress, resulting in the disruption of homeostasis and ion distribution in the cell. In regard to heat and drought stress, overall it was found that a combination of drought and heat stress had a significantly greater determinantal effect on the growth and productivity of these plants and crops compared to each of the different stresses applied individually. A comparison of all major U.S. weather disasters that exceeded $1 billion each between 1980 and 2012 indicates that a combination of drought and heat stress caused extensive agricultural losses of $200 billion. Several studies had been conducted on different plants and crops to evaluate the effects of drought and heat stresses combined. Results showed that the effect of combined stresses differs from a species to another. There is no generalization of the combined stress effect on all plants, but we can have a look at case studies and observe the effects on each individual species. In Arabidopsis, Weil et al. Dissected the effects of drought, heat stress, and their combination on growth rates. Plant growth was significantly reduced under both stresses, and the combination resulted in even more determinantal effect. The combined effects of these two stresses were generally additive, suggesting a certain degree of independence between the mechanisms regulating the responses of plants to drought or heat stress. Undeniably, some growth rates were altered specifically in response to one but not the other stress. Yet we can see an antagonistic response in the same plant for the same combination of stresses. An example here is that heat stress can cause an increase in stomatal conductance in order to cool the leaves, while drought stress causes plants to close stomatal openings. If these two stresses occurred at the same time in the plant, leaves would be much warmer, which shows an antagonistic response. Another studied model was barley. In barley, drought or heat stresses reduce plant growth with more severe effects as a result of drought. The combination of drought and heat stress reduce plant growth to a much greater extent than drought or heat applied individually, which indicates an additive effect just like in Arabidopsis. By contrast, the effects of heat or drought and heat combination on photosynthetic rate were more severe than the effect of drought on its own on this parameter. The combination of drought and heat stress could require antagonistic response of barley plants. For example, during heat stress, plants increase their stomatal conductance in order to cool their leaves by transpiration. However, if the heat stress happens at the same time with the drought, plants would not be able to open their stomata and their leaf temperature would be 2 to 5 degrees higher, same as in Arabidopsis. In addition, proline that accumulates in plants under drought did not accumulate during a combination of drought and heat stress. So this gives us an antagonistic response as well. Now let's have a look at chilling and drought stress. In a study conducted by Salas et al. On sugarcane, results showed that drought alone or combined with low substrate temperature led to a large impairment in CO2 assimilation and enhancement in the generation of excessive energetic pressure at the PS2 level, with photochemical activity being relatively preserved. The cold and drought treatment reduced the maximum quantum yield of PS2 in sugarcane by 10%. The reduced photosynthesis caused reduced plant growth. Shoot, root, and total dry matter production showed a decrease when subjected to water deficit either alone or combined with low temperatures. Compared to the reference condition, the combination of cold and drought reduced the total dry mass by 37%. Now let's have a look at heat and soil stress. The combined effect of reduced plant growth was antagonistic since growth rate reduction in salinity was much higher than it was in heat alone or in heat and salinity combined. As expected, sodium concentrations in control and heat-treated plants was insignificant as these plants did not receive additional sodium chloride. Sodium concentration was 38% higher in both leaves and stems of plants grown under salinity as compared with the salt and heat treatment. However, sodium concentrations in roots of plants grown under salt and heat treatments was 28% higher than those found in plants grown under salinity. Water potential values obtained from plants grown under salt and heat were similar to those obtained under heat treatment, with reductions of about 35% with respect to control plants. The lowest values for water potential were recorded during the salinity treatment. If we look at the three stresses combined, low temperature, drought, and soil stress, we see that osmotic stress is the common occurrence with all three stresses. Soil drought and to some extent cold stress causes an increase in biosynthesis and accumulation of ABA, which can be rapidly catabolized following the relief of stress. ABA has a large role in osmotic stress. This figure is a model for the role of signaling factors in stomatal closure and retrograde signaling during water stress. In response to drought stress, ABA stimulates a signaling pathway that triggers the production of reactive oxygen species, which in turn induces an increase in cytosolic calcium. As you can see through this figure, this is a very complex response which encompasses many ions, receptors, and different transcription factors to make sure stomatal closure is complete. So now you might think, what is the solution to avoid all of these damages? Well, as you know, weather is hard to control and predict for longer terms, so drought seasons can be scarcely avoided. Also, it would take an incredible amount of work and materials to fix and wash off the salt in acres of fields. Scientists decided to take a different approach in solving the problem and worked on increasing plants' hardiness to those stresses. Breeding of stress-tolerant crops is considered the most efficient strategy to maintain yield in stress-prone marginal land. Germplasm selection, marker-assisted selection, transcriptional profiling, metabolomics, proteomics, and transgenics are all techniques that are currently used for crop stress improvement. And now let's have a final look on what you should have learned in this module. You should know the answers to the following. What is drought? And name 3 plant responses to drought. What are the strategies plants use to tolerate drought? Name 3 adaptation and 3 acclimation responses in drought-tolerant plants. What is salt stress? And name 3 plant responses to salt stress. Name 3 adaptation strategies plants use to tolerate salt stress. What are the 4 patterns of response to stress and what is the difference between them? Give an example on each of the stress combinations discussed. And finally, what is the solution for avoiding these stresses? This concludes the last module of this course. Thank you for listening and good luck with your final exam. Goodbye.